Welcome, welcome. Good morning. Welcome everyone to this very special event, Richard SKALAK Bioengineering Lecture, hosted by the Department of Biomedical Engineering at Columbia University. So my name is Ed Guo. I'm the chair of the department for several months and at Columbia University. So very excited to continue the tradition of hosting this distinguished lecture, which began in 1996. That's where I started as a faculty at Columbia, and was named in honor of Columbia alumnus and pioneer bioengineer and then distinguished professor Richard SKALAK. So I print out a little bit of more information about Richard SKALAK. Richard SKALAK was a Columbian forever. After graduating from Columbia with a Bachelor of Science in Civil Engineering in 1943, he served in the U.S. Naval Reserve as an instructor in radar and sonar in Washington, D.C. 1944 to 46, returned Columbia after the end of the war, and then he served as an instructor in civil engineering from 1948 to 1954. But in the meantime, receiving his PhDs in applied mechanics, fluid mechanics in 1994, and then studied as an assistant professor. So he used to do a regular civil engineering do. I think it's pretty boring. For example, water hammer in a heating system. But later on in the 60s, he began to move into, I think, more important bioengineering. So he's one of the pioneers in biomechanics and biomedical engineering. And then coincidentally, this Sunday is 100th anniversary of Richard SKALAK's birth in 1923. So we're honored to continue this celebrate Dr. Richard SKALAK, especially this year. We welcome his family members who have joined us, as I know, today on Zoom. And I will let my colleague London to introduce today's distinguished Richard SKALAK Bioengineering Lecture.
Thank you, Ed. So it's really a tremendous pleasure to introduce Eric Wieschaus as the SKALAK Award Lecturer this year. Professor Wieschaus is the Squibb Professor of Molecular Biology at Princeton. He's my own personal science crush. And the winner of the 1995 Nobel Prize in Medicine. And of course, this is what most of you likely know him for. This tremendously influential genetic screen that was done together with Christian Nusslein-Volhart in the late 70s and early 80s, using the Drosophila or fruit fly embryo. This was influential for so many reasons, one of them being that it really brought genes and gene activity to the center of our understanding of embryonic development. In addition to that, they discovered many, many genes that in the 40 plus years since then have been identified to be important not only for the development of fruit flies, but vertebrates and invertebrates alike, and are also genes that are critically important in understanding even adult onset human diseases. And so this is, of course, sufficient and good for the Nobel Prize, but the reason that we've invited him here to give the SKALAK lecture is actually the work that he's done since then, where his focus has really broadened to understanding how that information that's encoded in genes can actually be harnessed by cells in transforming, and physically transforming an early embryo in this very precise way, in such a stereotyped way that the complexity of embryonic development can take a single cell and make it into a multicellular organism. And it is because of this that this multidisciplinary approach that draws from genetics and embryology and microscopy and physics and mechanics, focusing it on this biological question, that his interests and his influence really parallel that of Richard SKALAK. In addition to that, and the many ways that he has really become a visionary in the morphogenesis and developmental biology field, he has also trained over the years countless scientists that are now themselves leaders in this area. And I think the last thing I'll say before I hand it over to him is that what you'll likely see from his seminar, and you'll certainly see if you have the opportunity to meet with him during his visit today, is this is someone that, despite how accomplished he is and the 40 plus years that he's been so fully immersed in science, he still approaches biology and science with a level of fascination and curiosity that is both deep and genuine and truly inspiring to get to experience. So with that, welcome, Eric. Look forward to your seminar.
It's a pleasure to be here. It's an interesting and an exciting thing now at the end of the pandemic to begin to have the opportunity to visit other universities, talk to people, present science. It's one of the really essential aspects of science is its social nature, that we not only do experiments in an ivory tower and then discover things or think we discover things, but we go out and test our ideas by talking to other scientists. And this is actually one of my first real entries back into public lectures. So I was very excited about the opportunity to do this. As a biologist, many things that are fascinating in biology, but for me, and I think for many of us, one of the most amazing phenomena is the realization that every one of us in this room and all the organisms that are out that we encounter every day can often trace their existence back to this single point following fertilization where you had an apparently simple fertilized egg cell. And somehow that egg cell has both in its DNA and in its structure the capacity to develop into something as complicated as we, or as complicated as any living organism in this earth. And this is a phenomenon that ultimately can be described or reduced to descriptions of gene activities or various levels of control. One of the things that I have always found most fascinating about the process is that it's not just biochemistry and it's not just genetics or gene activity. There's a visual transformation of morphology that has to be, and underlying that transformation has to be a mechanics and a mechanical understanding of how structure form, how you reshape an embryo. The work that I'm going to talk about today is done in Drosophila embryos, and let's see here. These are actually two fly embryos. One of the fly embryos are one of the impressive features about development, particularly fly development, is that these morphological changes are extremely rapid. So these two, I fixed these two embryos. They happened to hit the fixate of about 15 minutes apart. And this is at what we would call the cellular blastoderm stage. The embryo has about 6,000 cells at this stage, about 80 cells along the anterior-posterior axis. All the cells look pretty much the same. And 15 minutes later, we can see obvious morphological transformations, foldings that are happening, or where cells on the surface of this blastoderm are being internalized into the embryo. We can actually follow that process now because we can follow the process of individual cells. And what you'll see in this movie, this process takes, this as a span of this movie, is about 8 to 10 minutes. Is that you look at individual cells, what you can see is that this cells will decrease their apical diameter and then gradually undergo this process of moving into the interior. Now, I made these movies some time ago. More recent, the cells that are going into the interior these are what are called mesodermal cells, which were going to be dedicated in the embryo to form internal structures like muscle. And so obviously having them out on the surface may be fine at the beginning of embryonic development, but you want to get them inside. And so the question is the mechanics of bringing these mesodermal cells into the interior of the embryo. And there's a stereotypical process of morphological transformations. This is a little optical cross-section of the embryo. Now you can see the very columnar cells, and over a period of about 5 minutes you get a groove. 10 minutes the cells move into the interior, and then eventually once they're in the interior they will begin to spread out over the surface of the ectoderm, having undergone an epithelial mesenchymal transition to begin the first steps of making internal mesodermal structures. Now, more recently, obviously, to understand things, you have to be able to see them. You have to be able to look at them. You have to be able to record. You want to be able to record behaviors. And what we've begun in doing in the lab is making use of three-dimensional light sheet microscopy and complex computer reorganizations to be able to track the behaviors now of individual cells. And you can see them better. We're now looking at the dorsal surface, the ventral surface, and the lateral surface of the embryos. And you can see, if you watch these movies, you can see the formation of this furrow. It's occurring simultaneously in the embryo with lots of other processes rapidly in the embryo. Because we can follow cells, we can actually identify individual cells. This should play. Okay. And track individual cells through this process. And because we can track them and give individual cells names and numbers, we can not only follow the cells through development, but we can start at an end stage of development and track them back to the blastoderm. And that becomes important because we know from other studies, and I'll mention these, describe these briefly, that at this early blastoderm stage, the cells are highly and very specifically programmed with individual transcriptional patterns. So we're able to, what the goal of this is to be able to look and define behaviors, look and define outcomes, and then trace them back to the trace them back ultimately to genetic expression patterns at those early stages. Whoop. Okay. So one of the things that we can do, and this is to, you've seen these cells moving in the ventral furrow, these mesodermal cells moving into the interior of the embryo. And you can track all the cells in the blastoderm and ask how many of the cells are internalized. And when are they internalized? There's a heat map with respect to time. And the ventral furrow cells, which we can't see from the dorsal view, this is a view where the embryo has been split open. So this is the ventral side of the embryo. These are the first cells to disappear from the surface. And then at about 10, maybe 2 to 3 minutes later, a second fold occurs, and then third fold. So cells are lost from the surface. Overall, in the first 40, 50 minutes of this gastrulation process, 50% of the surface of the embryo is internalized. And what that means is that since the embryo doesn't fall apart, that the cells that are left on the surface have to compensate for that loss. And using movies like this, we can begin to follow the compensation and the expansion of adjacent cells, how the embryo responds and compensates for loss of cells. And we know that there are different pathways. The cells begin to divide, and as cells divide, they round up. Even if they don't increase their volume, that overall decreases the height and increases the apical surface area. Also, the cells just appear generally to passively stretch. And I use the magic word there, passive, and stretch. And when you look at movies, and in this introduction, what I wanted to do is just play you some movies to try to get you into thinking about the process. You see morphological transformations, and our brains automatically look for what are the regularities, and then how do these things happen? Where are the forces being generated? What are active processes, and how do those active processes actually function? But are there what we would call passive processes, where cells in the embryo respond to morphological changes that are occurring elsewhere? Can we begin to plot out? If we really want to understand morphology, we have to be able to distinguish, we have to be able to identify where forces are, what generates forces, what's active, what's passive, what are the properties, actually even, what are the properties of the material, what are the properties of embryonic epithelial cells that allow them to have active force-generating properties or behaviors or passive responses, and how are those distributed, how are those spatially controlled, both temporally and spatially. So that's going to be the idea of this talk, is that we have this extraordinary embryo, a fly embryo. All embryos are extraordinary, but extraordinary that over a short period of time does these morphological transformations. And we want to understand as deeply as we possibly can what are the driving forces, how does this actually occur? And it's my belief that doing this in an early embryo is probably about the best place that you can, most advantageous opportunity for studying these phenomena, because it's rapid, it's simple, there's not a history, there's relatively few cell types you can hopefully identify just from morphology, and from the morphology of the cells, develop hypotheses that suggest underlying mechanisms. So, how's the embryo shaped? What's active, what's passive? How do folds form? Why do folds form in specific places? The answer, at a certain level, like everything in biology, processes depend on genes, and on the proteins that are encoded by those genes. And knowing the genes, particularly knowing the genes in Drosophila, gives us very powerful tools for analyzing the process, and that, we believe, provides us with the first powerful tool for being able to begin to approach mechanics. So in Drosophila, over the past 40 years, there have been extraordinary advances because of the genetic advantages of the organism to do mutagenesis, not only to do mutagenesis screens to identify genes that, when mutated, disrupt processes, but actually to identify those genes, figure out what their products are, describe their expression patterns, understand what their biochemical activities are, manipulate their expression, test genetic models for example, for the formation of this ventral furrow. In Drosophila, we know that the patterning in the embryo, and we're looking now at a cross-section of the embryo, the patterning of the embryo and the specification of mesodermal cells on the ventral side of the embryo depends on a gradient of a maternal protein called dorsal that provides some positional cues and defines these cells here as ventral in fate. Dorsal is a transcription factor and activates the expression of a set of two genes called twist and snail that are essentially mesodermal fate determinants. They are themselves transcription factors and register these cells as being mesoderm and these cells as being not mesoderm, ectoderm. So we make these established cell fates, and then almost immediately after the expression of these transcription factors that determine cell fate, the cells in the ventral side of the embryo begin to change their shape and move in discrete patterns. Now, if you look at the embryo in more detail and you follow these cell shape changes, one of the striking features is that cells initially start at this blastoderm. They're about maybe 5. They're very high columnar cells. They're about 35 microns high, about 5 to 8 microns across. And you can see here the initial stages of this furrow formation as a constriction in of the apical surfaces of these cells, becoming more extremely constricted, and then this fold is formed. The cells move into the mesodermal cells move into the interior of the embryo. This process is going to require some force-generating machine. We know from the studies involving looking at the cytoplasmic myosin 2, that if you look at this stage here, the accumulation of myosin, what you can see is that it's localized to the basal surfaces of the cells. And this is a historical feature of the way that the embryo undergoes a process of cellularization. Most of the myosin is here. But in response to the expression of twist and snail, on the ventral side, we begin to accumulate this contractile motor protein myosin on the apical surfaces of these future mesodermal cells. And it's the accumulation of that contractile force here that drives the reduction in the apical and the formation of this furrow. So basically, the process that we're going through is that you have cells that start out as columnar. You accumulate myosin on the apical surface. That leads to a constriction. We know that the cells maintain constant volume. The consequence of that constriction is that the cells become longer. You can see that here in the images. And then in a process that we don't understand, after this apical constriction is completed, the cells shorten again. And having a constricted apices and constant volume means that the shortening now drives the cell into this trapezoidal or triangular shape, and that produces the torque that drives the cells into the interior. So it's mechanically an easy to understand process. Obviously, there has to be a connection then between this myosin accumulation and the cell fate determination, the decision to be mesoderm. What we know, and this is from large numbers of genetic screens, is one of the peculiarities of embryonic development, but especially strong in Drosophila, is that the mother, when she makes the egg, puts almost every protein that you know about in the egg. All your major cytoskeletal proteins, all of the adhesive proteins, myosin motors, all of those proteins are supplied uniformly in the embryo. So to put myosin in one particular place or in one particular cell does not involve the transcriptional control of myosin. Myosin is there. What the embryo has to do is express in specific regions in response to these cell fate decisions factors, proteins, gene products, that will govern the deployment of myosin. So when you think about the embryo is this machine that's sitting there, everything is there, and to produce morphological change, what you have to do is cause the local synthesis or local triggering or activation of myosin in this region, and not in these cells, not in these cells, in this region of this cell, not in another cell. So that's the basic process for us, is that we can assume all the mechanics that we know from cell biology, all the features of these cell biological molecules. What we want to know is how are these processes triggered in specific regions, and then what is then use that knowledge to ask what are the mechanical properties or downstream consequences of triggering myosin in one place in the embryo and not others. So we know that I've identified in that twist and snail, which are transcription factors, bind to and govern the expression of a relatively small number of genes, specifically in the mesoderm, that have activities which will drive the accumulation or activation of myosin. The way that works, and hopefully this shows one of these downstream targets, a gene called folded gastrulation, which is a signaling, a secreted protein which drives through a receptor signaling pathway a phosphorylation of myosin. And we can see the accumulation of folded gastrulation in the ventral furrow drives the accumulation of myosin and the constriction of the cells. Now, the way the pathway works, and this will be just a little bit, that folded gastrulation is a secreted protein. T48 is an internal protein. They interact with a 7-pass receptor. So the mesodermal cells are secreting this in a certain sense to coordinate this myosin transformation, and through a set of G protein this G protein coupled receptor, ultimately activate a Rho kinase, which will phosphorylate the myosin light chain, lead to myosin contractility. And it's myosin contractility that's required to drive that accumulation and localization on the apical surface. Now, the interesting thing about this pathway, which is we genetically defined, we know all the components, have been worked on a number of labs now, is that folded gastrulation exists or is identifiable based on homology in Drosophila, but not in other, even in far insects, it's hard to find. T48 is also rare. The rest of the pathway is just this conserved machine that operates in cells. It's the same pathway that drives stress fiber formation in cell cultures. These two genes are also conserved. So fate determinations and the machinery to achieve morphological change are conserved. What animals do, apparently, is evolve in developing, as embryos have different shapes, they must localize under different conditions. The triggers that activate these pathways can be rapidly changing during the course of embryonic development. And these pathways are actually used constantly in fly development and triggered by a variety of different stimuli in flies. Also, these two proteins are also used again and again for any invagination in flies, but not in others. But their job is really just to trigger these mechanical events, the contraction of myosin. Now, one of the interesting features is say, okay, so it's all clear. You get myosin, you constrict the apices of cells, the cells become a smaller and smaller apical surface, and this is what drives the... what we had actually anticipated that to reduce the apical surface, if there was a band of the myosin would form some little band of myosin around the circumference, and the cell would, and the circumference would squeeze, and the individual cells would reduce their apical surface area. So we anticipated that the myosin that was accumulated would kind of outline individual cells on the apical surface and provide belt-like constrictions. Amazingly, when we looked at the accumulation of myosin, we saw something totally different in that this is the myosin accumulation. That myosin across this network of 600, 700, 800 cells that is mesoderm forms this continuous interconnected network that seems to span the cellular arrays. Now, obviously, the myosin molecules are not themselves spanning the arrays. Actomyosin cytoskeleton, through junctions, is forming an interconnected network over the whole surface of this mesoderm. And this would be a movie if it would play, but it won't, so hopefully I'm going to have trouble. All right, so an interesting, then the most telling feature of this network is that it's contractile, but it has a slightly more complex pattern of contractility. And that if you look at individual cells, maybe I will try escape here, or good. That you can follow myosin accumulation in an individual cell. You can see it forming and going away, and individual cells are pulsing in their myosin distribution, but they're all part of this network with that pulse accumulation of myosin in a given region that has about a 90-second periodicity. The myosin's pulsing in cells, and that pulse is different from one cell to the next to the next. So all these cells are undergoing this kind of pulsing stochastic variation, and they're contracting, and overall, over a period of about 6 minutes, the whole surface of the mesoderm contracts down. And I will... and you can follow it here in an individual cell where myosin accumulates and then goes away in that cell. And you can follow the shape of the cell, which is a polygon. And as myosin accumulates, the cell actually becomes contorted because the myosin fibers are perpendicular to the surface of the cell. The cell becomes contorted, and then when myosin goes away, it relaxes, but the size is less. And so this progression is a stepwise process that's coordinated with accumulation of myosin. Now, what we're doing here is just only looking at the apical surface, and if you looked at those the images from the SEM before, you look at other, you can see that lots of things are going on in the cells. The cells are elongating. Actually, the nuclei are moving from one place to the cell. The microtubule cytoskeleton is elongating. Total surface areas are changing. You have membrane incorporated. Lots of things are going on in these cells. What we see here is the apical surface corresponds to this pulsing pattern of myosin, and that as myosin accumulates, you correlate that with correlates with a reduction in apical surface. Now, what's actually more interesting is that when you go back and you look at any feature that's involved, any morphological change, be it elongation, or be it increase in surface area, or position of the nucleus, all of these processes in detailed analyses are also stepwise in their nature, and they show the same periodicity and the same correlation with myosin, with the myosin accumulation in individual cells. And so that, for us, was the strongest argument that maybe understanding morphology was something that could be simple. That it's all these, that you don't have to have lots of processes, that everything is coupled potentially to this constriction, this myosin activation. The fact that myosin pulses gave us a handle for viewing the process differently, and also a process that, an argument that all we need to do is to understand myosin and its accumulation, to understand, and then relate the rest of all the other morphology to mechanical changes in the surface of the cells. And so that's going to be one of the goals of this seminar, is to ask, is that reasonable? Can we begin to do that? What's involved in that process? So, can myosin accumulation on the apical surface drive all aspects of cell shape change? How can we ask that question? So, one of the things that you'd like to do is to have other markers. If you think about many of the aspects of a cell elongation, essentially involves redistributions of cytoplasm. So one of the things that we wanted to do was to develop ways of marking cytoplasm and following cytoplasmic flows in cells of embryos. And the strategy that Bing He and Konstantin Dubrovinsky developed was to take fluorescently labeled beads and inject them into embryos. Inject them in at actually an early stage before the embryo had fully cellularized. So these individual beads would be just incorporated into the cytoplasm of the embryo. And then when the embryo was partitioned into cells to form a cellular blaster, individual cells would have their cytoplasm labeled. Because the cytoplasm is viscous enough, the beads follow the movement of the cytoplasm. We can record the movements of the beads and reconstruct the flow patterns in the cytoplasm. And so possibly we can we'll go, and so now we can reconstruct. And so the patterns that you see are that there's a flow overall of the beads in a kind of a reverse fountain kind of flow, where beads come and flow down. And I'll point out to you that this flowing down is essentially the equivalent of the cell elongation, the nuclear displacement property, all of those morphological phenomena that we see in single cells. And if one wants to model or think about it in a cartoon-like way, we can think of this just as a contraction of a laminar, basically laminar flow. As you have on the surface of the embryo myosin accumulation, you accumulate, drag the surface, and drive a flow pattern, which is like the one that we see, cartoon conceptually. Now, one of the things, that was a cartoon, I drew that cartoon. Other people in the lab do math. And one of the remarkable things, though, at Constantine, showed that if you looked at the patterns of flow of these beads, and you can predict them using the Navier-Stokes equation, basically the Stokes equation. This means that you would predict the flow. Essentially, if you know the behaviors out on the outside perimeters, you can predict the behaviors of the flow. And then you can ask, can we describe the flow patterns, these global flow patterns that we see? Are they compatible? Are they driven by the same, by processes that would describe Stokes equations for a laminar flow of a viscous fluid? And this is amazingly, there's very little deviation from that. That is gratifying, but it's also surprising, because I told you that we're looking at beads in cytoplasm inside cells. And we're looking at an array of 30 cells, and the flow of cytoplasm, we're saying, is continuous through this epithelium, and the Stokes equations work when you're dealing with tiny, tiny little minuscule, they're big objects or cells or cell membranes should block or occlude that process. It's not, and yet it doesn't seem to. And so what that suggests us is that our focus on cell membranes may be essential for isolating gene activities from one cell to the next, but that at least the lateral and basal cell membranes don't have very significant mechanical properties. So to test that idea, what Bing did was to use wheat germ agglutinin and then try a different strategy where he injected the beads in and had them incorporate on the outside of cells on the membranes as they were forming. And then did the same analyses and asked if membranes flow differently than cytoplasm. And the answer is no. Everything flows together. And with it, we're beating a dead horse maybe by this point, but it's that these it doesn't make any difference in the behavior of the bead, whether it's in the cytoplasm or on the surface. That membranes, at least these, offer very little resistance to the flows and the mechanical properties that we're observing here. So that means that, well, are membranes important for anything? Now, that would be a crazy question to ask, because we have an embryo with 6,000 cells, and each of those cells is separated by. But is the subdivision of cells necessary for morphogenetic processes in the fly embryo? And this isn't a question that you would easily be able to ask in anything other than flies, but it turns out it was something that we could address in flies because of a peculiarity of Drosophila development that I've kind of glossed over. Drosophila begins its development fertilized egg, and then undergoes a rapid series of nuclear replications and cleavage division, where the nuclei undergo these synchronous 13 rounds of division until they reach what's called the cycle 14 blastodon. Those because fly embryos have a premium on developing as rapidly as possible and being the first little larva to get out of the egg and hatch, and they do all this incredible stuff in 18 hours, one of their strategies for speeding up early development is to allow nuclear replication, but not go through the mechanical process of subdividing the embryo into 2 cells, 4 cells, 8 cells. Instead, they replicate their nuclei up until they have 6,000. And then, when they reach 6,000 nuclei, they're at what's called a syncytial blastoderm. They stop dividing, turn on gene expression. This is the point when twist and snail come on significantly. And turn on a set of genes which are involved in cellularization and cellularizing this syncytial embryo into the cellular embryo. And that process whereby membrane is made and inserted between, and membrane is pushed between adjacent nuclei to form this epithelial cell layer, this new epithelium that just formed and is going to do these tricks for us, it requires 2 zygotically active genes that we know. That one is called DUNC, and one is called discontinuous nuclearase, one is called SLAM for slow as molasses, and neither one by itself knocks out or eliminates cellularization. But if you eliminate both of them together, you make embryos which are perfectly normal up to the beginning of cycle 14, have all their nuclei, the dorsal gradient, the twist response, but now but are unable to subdivide into 6,000 cells. So you have one large 500 micron cell with an array of 6,000 nuclei on the surface, each of which is transcriptionally active in different ways that program different regions of the cytoplasm to enter into this quasi-mesodermal fate. The slam dunk phenotype is, so you can see this here. This is a wild-type embryo. It's completed cellularization. This is in cross-section. Here you can see the expression of twist in the ventral region. This is a slam dunk. Remember, they have the membrane. What they're unable to do is organize this membrane into coherent sheets. So it's a very floccular membranous material. And they don't cellularize, but they do express the appropriate genes ventrally, twist and snail. They activate myosin and will drive. Is that better? Okay. And so we can ask, do these embryos gastrulate? Cup half empty, cup half full. They, amazingly, they do. So this is the normal gastrula. This is the mesodermal cells moving in. This is the cytoplasmic flow that is elicited in this single large-celled embryo. We can do the bead experiments, measure the flow. The flows are, in fact, the same. And so what that means is that to do the first steps, these aspects of gastrulation, we can do, you don't need cell membranes. What Bing has actually shown is that if you plot the redistributions of cytoplasm that occur in these cases, you mimic the cell shape changes that occur in individual cells. So you can reconstruct the outlines of individual cells just by the flows of regions. If you take a region of the cytoplasm in this fully syncytial embryo, say this block is a cell, how will it transform? And the shape transformations that occur in those domains of cytoplasm are the same. But it's not good to be a big cell with a big membrane with no cell membranes. And about 25, 30 minutes into the process, the mechanical stresses on the embryos, and they begin to collapse or tear themselves apart. But even before they ultimately tear themselves apart, if you look at them, you see that they can't actually do everything that the real mesodermal cells can do. normally, if you follow here, we're just looking at nuclei, we can see this displacement of the nucleus, and, here, this is wild type, sorry. The flows of the cytoplasm and displacement of the nuclei, and then a furrow formation and a movement, an invagination of the furrow, which remember is associated, whereas in the single-celled embryos, they can do the apical constriction, drive a laminar flow, drive a redistribution of nuclei, but they can't do the actual invagination. So we go back and look at the process. There was two stages, the way I described it. There's this initial elongation, and then there's this shortening phase. And the shortening phase was something that was interesting and confusing, because unlike the shortening phase, unlike the first phase where we could see myosin accumulating, we had candidate molecules. When we look for anything that's special in the shortening phase, it just happens at maximum constriction, then suddenly, and the system goes back and shortens and drives the cell shape change in the internalization. And we've been unable even to find mutants. We can block myosin accumulation or folded gastrulation blocking the first phase, but we can't block the second phase. So there's something mysterious then about this second phase and this invagination process, of this shortening process, that we don't understand. So what I'm going to do next is give you what's probably the most single controversial model we've proposed in mechanics of cell shape change. I'm embarrassed. I'm going to do it because I love it, and it's just challenging. And I'll show you. I'll work through our logic here. Now, the observation came from something originally done by Matthias Kaschuber, that if you follow this process, the cell volume is constant. If you look back in the normal case, and cells elongate during apical constriction, but they return to their original length. So they elongate and then they shorten. And so what we started thinking about was the possibility that the shortening is actually an elastic response to the elongation, to apical stretching, as though you had springs that would be stretched and then spring back. I thought that was a great idea, but then someone, I think it was Matthias or Constantine or something, pointed out that if you look at turnover, if you use FRAP experiments, fluorescently labeled cytoskeleton or components, and ask what are the turnover rates for cell biological components, for E-cadherin or any cytoskeleton, they're on the second level. You turn over you're turning over, when you measure things in general, the lifetimes are very short. And we're talking about a spring, if you will, that we want to extend for 10 minutes and then drive a shortening. So in terms of the, if any of us have had spring toys as children, what usually happens is that the springs, which initially have some fixed length and will return to that length, they will, because of restructuring, breaking of chemical bonds within the spring, the spring no longer returns to its length, so the springs relax. And that depends on molecular restructuring, and I've just told you that the cytoskeletal components that we know about have lifetimes that are on the order of seconds, and this is why we think of cytoplasm as being viscous as opposed to elastic. So we decided we would go out and see if we can test the idea by trying to, measure elasticity in fly embryos during gastrulation. This is, so we built the little electromagnets and injected magnetic beads, and actually more efficiently ferromagnetic fluids that form little balls, and inject them into the embryo and ask, can we detect elastic responses, and if so, what are the lifetimes of the parent springs involved, and can they work on the order of that? So hopefully, yeah. Okay, we're going to inject here, and now if this movie, I might need to, it was a movie. Yes. Okay. So you can see that's the magnet coming in, that's the little droplet, and what happens is the droplet is elongated, and then when you pull the magnet, the droplet condenses, but it doesn't move back to its original position. So that's what you expect for viscous fluids. It's that they'll be displaced, and then everything stays where it is. In this experiment, what Konstantin's done, he's done these on these during these syncytial or acellular stages of mutants, and he's put the bead, the droplet, in the middle of the embryo and moved it through the cytoplasm. So in these acellular, these embryos, everything appears as viscous. In the next experiment, what he did was actually embed the cytoplasm, embed the droplet into the cortical cytoskeleton associated with the membrane. And now what you can see is a very different behavior. Put the magnet there, pull the magnet away, and everything comes back. So there are elastic places in the cortex of the embryo. It does have this elastic response. You can do a chymograph. You can show, let's go back. There. The displacement and the return. That's just visually. You can plot it out. And what you can see is two things. Actually, you can use these curves to calculate the spring, the lifetime constant of the spring. You can actually do it from these values, the slope here, putting a spring under constant tension, or you can do it from the return, because as you notice, under the experiments that we're doing here, the droplet doesn't return exactly to the same place. So we can use this data to calculate spring lifetimes of these springs. And in one set, in the set of experiments, if you just treat the data like this, what you find is that you have lifetimes of 4 minutes. 4 minutes is longer than you would have expected from the known cytoskeletal elements. So there is elasticity, but it wasn't really quite long enough to do what we wanted it to do during gastrulation. And we're not really measuring in gastrulation, we're just measuring the cortical tension here. But what Konstantin then realized was that, in part, this process is dependent on, it can be, what this elasticity depends on is, it depends a little bit on what you're pulling. If you have a matrix which is stiff and you pull on it and extend it, then all of the extension comes in stretching the individual spring components. And when you release it, a stiff thing will spring back if it's elastic. Networks can also be arranged in what we think of as a floppy organization, where you extend them, and the part of the extension is involved in the preferred bond angles, or the interface angles here. So you can stretch, and depending on what's elastic, whether the springs are elastic but the bonds aren't, you can display, you will have a different, a lower elasticity in this case, because you're distorting the shape of the network as well as stretching the springs. So you can, to test that idea, what you can do is pull twice on a network. Pull once and release, and then pull again. And if this is occurring, then you've reoriented and you're really measuring now. This allows you to measure the lifetimes of the springs. And when you do that, we get things 9 minutes. 4 minutes and 9 minutes, we're both in the right ballpark. 9 minutes is better, a closer fit. And what it suggests to us is that it may be misleading for all of us as biologists to think biological systems are essentially viscous. And when you do that, what you do is you look at a morphological change, you imagine everything is viscous, and you say, we have to put these forces, these motors here to transform this shape into another shape. So every event involves an active burning of energy and a transformation from here to here. And we're good at that. We do finite analyses. We can figure out how to transform morphologies by constantly doing work. The idea of springs, and why I'm still struggling with it, is that you do work once, biochemical work once, and you store some of that work as potential energy that you use for a subsequent process. And I don't know how common that is going to be in biology. I think it's an interesting opportunity that we have to think about. We haven't thought about it very much, and there's lots of things that are worth thinking about. It's not clear even, I think this is a good theoretical problem. If you have biological material, therefore, that you imagine is composed, of many components, and on some time scale, every one of those components is elastic and then becomes viscous. So any type of transformation, everything is like springs. Some of the springs have millisecond lifetimes, and some of them have minute lifetimes. So when you measure properties, though, how do those lifetimes add up? And how can we think about, if there are many springs which are short-lived and a couple of long-lived springs in a system, what are the constraints on the long-lived springs? Are the long-lived spring, is the longest-lived spring the constraint on the system or not? Or how does one control that biologically? So that's the exciting thing about experiments. Always, you can get something, you can get some interesting results, and then you realize that in the best of cases that you don't really understand, there's more things that you really have to think through and decide. So I'm going to stop at that point. A relaxation times t, and then can we regulate relaxation times for springs? And yeah, those are. So this was a fun talk for me because I decided I wanted to kind of globally review the kinds of morphological studies that we've done, both using genetics and using more mechanistic, mechanical based approaches, and try to put it together and then try to point out where things we know, things we don't know, where we're efficient at knowing things. And I think the powerful tools of molecular biology and genetics puts us in, for certain aspects of the pathways, puts us in a really good position for understanding things. But it seems to me that that we don't have really good handles, or our handles for morphology and mechanics are still largely morphologically based rather than experimentally based. It's very interesting to go back to the movies that we're making now with tumors, where you see stretches of cells and see behaviors and try to begin to actually correlate behaviors, build models of what's active and passive, and then go back and try to test those genetically by local perturbations. So that's where I think the field is right now. It's an exciting time to be a scientist, it's exciting time to be a geneticist, it's an exciting time to be a biomechanics, someone who runs along with the biomechanists. So I'll stop there. Thank you.
Thank you, Eric, for a really exceptional and very eye-opening seminar. Really enjoyed it. We want to present you with this plaque in recognition of being the Richard SKALAK Ward Lecturer of 2023. So thank you again.
Thank you.
Okay, and so we have a time for a few questions. Just raise your hand. I'll bring the microphone to you.
Hi, Eric. Beautiful talk, as always. I had a question, both in the kind of movies you showed at the beginning of the cell tracking with Tomer, and then also in the ventral furrow. There seems to be kind of remarkable coordination between left and right sides of the embryo in all these processes. Can you comment on kind of where that comes from?
Where does the coordination come from? We've actually spent some time actually measuring even processes like germ band extension, where the right side and the left side their behaviors are basically involve cell movements and intercalations and random story. In total, even though everything seems stochastic and not predictable when an individual T1 occurs, in total they add up to be remarkably similar. So the question we don't understand is whether that's something that can just happen because we add up enough things and average out, each of which has a limited variability, or whether there's something that really coordinates. What is odd is that even though wild type under the most delicately treated conditions does it perfectly fine, if you mistreat or mangle your embryos, they will begin to show right-left asymmetries, and many mutants will do that as well. And so what that suggests is that it can't, and yet we're not really shifting averages of behaviors there. So it argues that there is something that either coordinates or writes things. Beyond that, I don't know. It's a good question.
Other questions?
Yeah, thank you for a terrific presentation. So you mentioned that in the single cell mutants that there is some degree of apical constriction takes place. I wonder if you could say a little bit about that, because presumably the apical networks are not there in a normal sense, right? Because there's no membrane. But
yeah, it is. Yeah, there is a plasma membrane on the surface of the single cell. So there's a cortex. That cortex has a actin-myosin cytoskeleton associated with it. So what we're doing, actually, I think, is a process that maybe is similar to what would happen on a smaller scale in a single tissue culture cell in culture, which can locally, in the cortex, develop a myosin constriction, a local little laminar. I cut the movie where we actually focus down on those single cell embryos and look at myosin, and you can see myosin now streaming in the cortex, focused on the cortex, streaming and driving this movement of the cortex, and underlying cytoplasm then being driven by viscous flow. So when we say they're acellular embryos, they're really one cellular embryo. It is interesting, though, that they have the cortical properties to be able to respond to folded gastrulation, phosphorylated myosin, everything to develop a local constriction. I love those embryos. They do some things, and they don't do germ band extension very well. They don't do it at all, actually. But those are all processes many processes require exchange of membranes or movement rather than flow. Even though we can describe them as flow, the driving forces are probably changes in adhesion, and those processes don't occur in the single-celled embryos.
Maybe one last quick question. Huh? What's up?
Good.
Oh, That's all right. We can close there. Let's thank Eric again for a really great seminar.
That was really great.